Hi everybody and welcome to this lesson. So this week we are going to be looking at key area 2.6 which is the environmental control of metabolism, and we're going to split this down into three lessons. So the first lesson, which we'll look at today, is looking at growth media and environmental factors. So what materials and conditions do we need to grow a microbe? The second lesson is going to look at phases of growth, and then the third lesson is going to look at analysing those different phases of growth and analysing the growth of microbes. So in this lesson, it's quite nice and straightforward. We're just going to look at how do we grow a microbe. So what are the conditions that are needed to culture microbes, and then what are the materials that we need if we're going to culture a microbe. So microbes is a really broad term. There's thousands of different microbes. Some of them are prokaryotic cells, so very primitive cells like a E. coli bacterium. Some of them are archaea, like our thermophilic bacteria that live in very hot vents in the bottom of the sea, and then some are eukaryotic cells, so things like yeast and penicillium. So there's a wide range of different microbes, and therefore they all have different requirements for growth, and it's important that we cater for each individual microbe depending on what we are culturing. So why do we actually use microorganisms? They're very easy to culture. They reproduce and grow very quickly. Their food source is very cheap, and they make useful products for us. So for example, antibiotics and hormones, and obviously in National 5 you looked at genetically modifying bacteria to produce particular products like insulin. So there's all that as well. And we can manipulate their metabolism, so we can get them to produce the products that we want, and we'll take a look at that later on in this key area. So what are the requirements for the growth of microorganisms? So microorganisms, if they're going to be producing something that we're going to use as a product, they need to have the materials to produce that. So they'll need things like amino acids to produce proteins. They'll need vitamins, fatty acids, and they're also going to need energy for growth. Now, some microorganisms can actually produce all of these things themselves, like the amino acids, the vitamins and the fatty acids. Some of them will need to take them from their environment. The energy source that the microbe is going to need is either going to be coming from the light, if they're photosynthetic organism, or it would be provided by chemical substrates within the material that we're growing them on. And what we grow them on is something called agar, and agar can come in lots of different forms. So it can come in a liquid broth form, or it can come in a solid medium agar, which you probably recognise from lessons. So we pour the liquid agar onto a petri dish and allow it to cool, and it forms like a solid sort of jelly-like surface that microorganisms can grow on. Obviously you're restricted in terms of the space when it's a solid agar, whereas with a liquid broth, because it's sort of three-dimensional, you can grow a lot more microorganisms within it. And you know, you can have it on a petri dish, which is quite a small surface area, in a bottle, or if you're growing things on a massive scale, then you'd put that liquid broth in a really large stainless steel fermenter, and then all you need to do is you need to make sure that you're giving it the ingredients that the organism requires. And there is a lot of different types of agar depending on what microorganisms you're growing. So each of these different agars, the reason they're different colours is because they all contain slightly different nutrients, and so it's important to select the correct one depending on what you're trying to grow and what sort of stage of growth you want that microbe to get to in order for it to produce the thing that you're after. Now, we've talked about the materials that a microorganism needs in order to be cultured. The other thing we need to think about is the conditions that a microbe needs to be having in order for it to be cultured. And one thing that we're going to cover is something called aseptic technique. Aseptic technique, the reason for it is twofold. So one reason why we need to carry out aseptic technique is to prevent contamination of the microbe we're growing with another microorganism, because otherwise that means that that microorganism that's contaminating your agar is going to be competing with your microbe for resources, and therefore it would reduce yield. So we don't want that to happen. The other reason is if the microbe that you're culturing is particularly dangerous, then we don't want it contaminating the environment. So it's really important that we carry out techniques that stop that microbe from exiting where we want it to be grown, and it causing harm to human health. So in the Google Classroom, I have put a little video clip of aseptic techniques in the biology lab. What I want you to do is I just want you to have a little look at that and identify 5 different methods or 5 different features of aseptic technique. So 5 different things that we could be doing in the lab that prevents these 2 things from happening. If you're growing on microbes on a really, really large scale, and what you would use is you use a computer control fermenter. Now the computer control fermenter provides aseptic conditions, so it's a completely sterile environment. Like I said, you can grow a microorganism on a really vast scale to produce useful products. If you're wanting to produce something like antibiotics or even the insulin that we're talking about in that National 5 from our genetically modified bacteria, you would use a computer controlled fermenter. And they hold thousands of litres of nutrient liquid and therefore can produce huge quantities of the product that you're after. Within the fermenter, there are sensors that will detect the environmental conditions. So the computer will the sensors will sense and feed back to the computer things like the temperature, the pH, the oxygen and the glucose concentrations. And if they vary at all from what they should be, then the computer will respond by adjusting those particular conditions to make sure that that microbe is kept in the ideal conditions for growth. So it has all of the materials it needs within the agar broth, and it will have all the conditions it needs met by this computer controlled fermenter. When the conditions are appropriate and the required product is released from the microbe into the surrounding medium. So when the conditions are absolutely perfect, your microbe is producing that hormone or that antibiotic that you need. When you've got the necessary volume or concentration, the computer halts the process, and that saves energy and prevents raw materials being wasted. Now, what I'd like you to do from this lesson is to make notes on the conditions and materials needed to culture microorganisms, and then watch that little video clip that I've posted in the classroom on aseptic techniques and make a note on 5 different methods. So remember, what we're covering today is what the conditions are needed to culture microbes and the materials needed to culture a microbe. So good luck with that.